can happen. So depending on the levels of the FGF in the AER, the ZPA can be either activated or repressed. So let's say we have a lot of FGF expressed in the AER. Okay, when we start having this high FGF, we'll have a negative feedback loop that will happen. This negative feedback loop is called FGF gremlin 1 loop. This will repress gremlin 1 to repress the activity of this protein. And I remind you that usually gremlin 1 prevents BMP from inhibiting the FGF in the AER. So in this case, this negative feedback loop will inhibit gremlin 1. So this will activate the BMP in order to inhibit the FGF, and this will decrease the levels of the FGF8 in the AER. And on the other hand, when we have very low, when we start having very low levels of FGF, 8, 4, 9, and 17, etc. So I'm saying FGF in general. When we start having low levels of FGF in the AER, this will induce a positive feedback loop called FGF sonic hedgehog loop in order to activate sonic hedgehog in the ZPA center. So sonic hedgehog is activated, so it will induce the expression of gremlin 1. This gremlin 1 will prevent BMP from inhibiting FGF, so that this will increase the levels of the FGF in the AER. So now I will give you an idea about the correlation between the different axes during LIMS development. Wnt7a, as I explained before, it's important for the dorsal ventral axis growth of the limbs. So mice deficient for Wnt7a lacked not only the dorsal limb structures, but they found that also posterior digits are lost in these mice. And this, of course, will suggest that Wnt7a is also needed for the anterior-posterior axis. In other experiments done, they have seen that the dorsal ectoderm, when it's removed from the limbs under development, this will result in the loss of the posterior skeletal element of the posterior bones. Okay, so the reason these limbs will miss the posterior digits, or they will lack these posterior digits when the dorsal ectoderm is removed, is due to the fact that sonic hedgehog expression, the main active agent of the ZPA, sonic hedgehog expression, was greatly reduced in this case. So when they tried to bring back Wnt7a by inducing its expression by a viral induction, this was able to replace the dorsal ectoderm. So the dorsal ectoderm was restored, is again present, and this also restored the sonic hedgehog expression. So they found that the sonic hedgehog start to be expressed again, and this restored the or rescued the posterior phenotypes. And of course, this experiment showed that the expression of sonic hedgehog is stimulated by Wnt7a. So it seems there is a correlation between the dorsal ventral axis mediated by Wnt7a and the anterior posterior axis controlled by the ZPA and the expression of sonic hedgehog. In a third experiment, they have seen that when we overexpress Wnt in the dorsal ectoderm, this causes an overgrowth of the AER and extra digits can appear. So what does it mean? It means that the proximal distal patterning, okay, controlled by the AER, is not independent from the dorsal ventral patterning controlled by Wnt. So there is a correlation between the dorsal ventral axis and the proximal distal axis. So according to these experiments, we have seen that we have during limb growth correlation between the three different axes of growth. So now there is a very important question asked: how embryonic limb development is stopped during the growth? Okay, so of course the limbs cannot grow to the infinite, so they should be stopped somewhere during development. So how this can happen? So so during the growth, you know that the ZPA produced sonic hedgehog. Sonic hedgehog induced gremlin 1. expression. So this gremlin 1 will prevent BMP from inhibiting FGF in the AER, so that the FGF will be expressed and the AER will be maintained in order to promote the growth of the limb according to the proximal distal axis. But during the growth, as the limb bud expands and increases in size, the gremlin signal will fail to reach the ectoderm. So its activity cannot be that efficient, so since it cannot be diffusing over large distancing to inhibit the AMB, to prevent the BMP from inhibiting the FGF8. So it cannot have any more an activity or an effect on the BMP. So in this case, BMP will be free. It cannot be inhibited by gremlin 1. So it will be active and it will prevent FGF expression in the AER. So in this case, no FGF in the AER. So it means that the AER will collapse and the ZPA, okay, is terminated and it will collapse also, since no FGF in the AER to support this ZPA signaling center, so that the embryonic phase of limb development will be terminated by this way. And now I will explain you the formation of the digits in two organisms, in ducks and in chicken. You see there is a difference in terms of cell death between the digits, between ducks and the chicken, and this happens during embryogenesis. So the digits are separated by a kind of webbing in the ducks, and this a kind of adaptation for the aquatic environment where these ducks can live. And at the opposite, it's not the case in the chicken. For example, here we can see a chicken leg, but usually the digits are completely, or they are separate, and we don't have a kind of webbing between these digits, since we have in chicken a necrotic zone between the digits. So it means that this membrane which is formed, it will be degraded during the chicken leg development in order to degrade this webbing, but this webbing will remain present in case of ducks. So in ducks we have minimal cell death, while in chicken leg we have extensive cell death between the digits. So in addition to this interdigital necrotic zone present between the digits, we also have other regions of the limb which are sculpted, or they will have a specific morphology induced by the death of the cells. For example, the ulna and the radius, the two bones of the arm, usually they are separated from each others by an interior necrotic zone that will induce the necrosis of the cells, and this will separate between those two bones, ulna and radius, to become two separate bones. And two other regions, okay, we can see interior necrotic zones in both cases. In addition, we have the anterior and the posterior necrotic zones, especially in the case of the chicken. And this will further shape the end of the limb. Okay, what's common here is that we have interior necrotic zone in both, but we have usually minimal cell death in the duck, since we have the webbing that will remain between the digits, while we have this interdigital necrotic zone very prominent in the case of the chicken leg development. So this is the skeletal pattern in tetrapods. It's formed by stylopods, zygopod, and otopod. Now we'll talk about sculpting the otopod, usually formed by the wrist or fingers for the hand, and ankles and the toe for the legs. So here we are taking the chicken hind limbs, and here we have the duck hind limbs. So we looked in both organisms at different markers like BMP and gremlin. So for example, in situ hybridization shown that in the chicken hind limb, but also in the duck hind limb, we have BMP expression in the interdigital zones between the digits. You see, this is the zone of accumulation of the BMP in chicken, but also in the duck. In addition, another marker has been observed. It's the gremlin. Okay, gremlin, it's known to be a protein that will inhibit the BMP. Okay, gremlin is a BMP inhibitory protein. Okay, it's by in situ hybridization. We can see it here in dark brown. Okay, pointed by these arrows in the duck hind limb. Okay, but in the case of the chicken hind limb, we can see that we don't have any gremlin expression in the interdigital zones. So when they looked at the apoptosis or the cell death in these interdigital zones in both chicken and ducks, we can see that we have in the interdigital webbing of the chicken, we have lots of cell death. These very tiny small red dots, okay, they present the death of the cells in the webbing or in the space called interdigital space. Okay, so in the chicken foot we have interdigital apoptosis, but the duck foot does not show any apoptosis. That's why we have this kind of webbing that will remain, because we don't have apoptosis in the webbing between the digits in the case of the duck, but we have it in the case of the chicken. So how this can happen? It seems like gremlin will inhibit BMP, okay, in the case of the duck, so that apoptosis cannot happen. Since BMP is a main inducer of apoptosis, BMP is active in the case of chicken, and this will induce the apoptosis and the removal of these interdigital zones between the webbing, between the digits, so that the webbing will be removed. So in the case of chicken, we have seen that we have inhibition of cell death between the digits by inhibiting the activity of the BMP factor. In this experiment, we can see the controlled chicken limbs, so we don't have any webbing between the digits. And here we have a chicken limb treated with gremlin, and gremlin is an inhibitor of BMP. So if we inhibit the BMP by introducing beads expressing gremlin in these interdigital zones, okay, we can see that the webbing will remain present between the digits and it cannot be degraded, since BMP activity will be stopped by gremlin, so that the apoptosis will be inhibited and we can see the webbing. So what's inducing apoptosis between the digits at the level of the autopod is BMP. We have lots of BMP factors like BMP2, 4, 7, etc. All of them are expressed in these interdigital zones. And when we block the BMP, this will prevent interdigital apoptosis. One of the BMP inhibitors is gremlin, but it seems like we also have another factor called Noggin. Okay, Noggin factor is also a BMP inhibitor. So for example, if Noggin, which is having a similar activity as gremlin, if Noggin is expressed throughout the limb bud, no apoptosis can be observed in this case, since Noggin inhibit BMP, so that it inhibits the apoptosis. So we have seen that BMP will induce apoptosis at the level of the autopod, but also it can induce the formation of the cartilage. Okay, the cartilage which is usually present at the articulation between the bone. This is called the articular cartilage. How this can happen? So it seems like BMP, as it has been found recently, can play two roles. It can induce apoptosis, but also it can induce the formation of cartilage between bones. So in the presence of FGF from the AER, BMP can induce the activation of one gene called DCOP or DKK1 in order to activate apoptosis. Also, BMP in the presence of Wnt or beta-catenin, which is the transcriptional factor of Wnt pathway, it can activate the formation of cartilage. But to prevent the effect of Wnt on this pathway, usually when the apoptosis pathway is activated, DKK1 will prevent the activation or the activity or the effect of Wnt on it in order to activate on the apoptosis and prevent the formation of cartilage. It means that we cannot have both processes exactly at the same time. So the same BMP can induce cell death or the differentiation into cartilage depending on the age of the target cell. How this was explained, according to this experiment, this has been explained. So during early limb bud stages, it means before the formation of the cartilage, they introduced beads These beads can secrete BMP factors, BMP2 or BMP7, to cause apoptosis. So, 2 days later, when they repeated the same experiments by introducing these beads to secrete BMP, the same beads caused the limb bud cells to form cartilage. It means that the apoptosis will happen before the formation of cartilage. And this is logical, since and it means that the webbing usually will be removed between the digits, okay? And once this webbing is removed between the digits, okay, this cartilage is formed by the same BMP pathway. So this is everything regarding this chapter about limbs formation in tetrapods.